We're ready.
Should we go or, okay. Good afternoon. Welcome back from lunch. Thanks for all of you for being here. It's really my great pleasure to introduce Dr. Paolo Casali, who's going to kick off our afternoon session today. Paolo is actually my former chair and a distinguished colleague, and is currently the University of Texas Ashbel Smith Professor and Distinguished Research Professor in Microbiology, Immunology, and Molecular Genetics and Medicine. Paolo's career and his contributions to the field of B-cell biology in particular are legendary. He did his early training as an MD in Italy, and then moved to the States and started an incredible career. Before joining UT Health UT Health San Antonio in 2013 as chair of the Microbiology and Immunology Department, Dr. Casali was the founding director of the Institute for Immunology at the University of California, Irvine, where he also served as Senior Associate Dean for Research and Graduate Studies in the School of Medicine. Prior to that, he was Professor of Pathology and Immunology and Director of the Division of Molecular Immunology and Director of the Graduate Program in Immunology at Weill Medical College, Cornell University, and Memorial Sloan Kettering. His research is focused on B lymphocyte immunobiology and the molecular genetic mechanisms underpinning the maturation of antibody and autoantibody responses, with emphasis on somatic hypermutation and class switch DNA recombination, plasma cell differentiation, and the generation of memory B cells. Paolo is credited with discovering human B1 lymphocytes and their role in the production of polyreactive antibodies. He also pioneered the construction of human monoclonal antibody-producing cell lines of predetermined specificity and isotype. I still remember the paper in Nature from when I was training. He's not that much older than me, though, honestly. His amazing career. His recent work has progressed to the study of epigenetic factors in the maturation of antibody responses. And so to address the in vivo epigenetic control of maturation of human antibody and autoantibody responses, his lab has recently perfected the generation of a new humanized mouse model, the description of which was recently published in an exciting and impactful paper in Nature Immunology. So today he's going to tell us about this exciting new humanized model and its many potential applications. Paolo, thank you so much for being here.
Thank you very much. Much undeserved. Much undeserved. Yeah. And now I have to live up to it. And yes, I'm quite older than Mike, but he's a young man. So, I hope I do this right because I'm not a PC guy. Let's see, how do I advance it? Oh, okay. Thank you. So why humanized mice? Well, the reason the application, potential application of humanized mice are quite obvious. All kinds of things from vaccine development to tumor immunology, immunotherapies, etc. And in fact, the first mice, immunodeficient mice, those were RAG immunodeficient mice, were injected with human peripheral blood mononuclear cells from HIV patients in the late 80s in La Jolla to attempt to model the infection, the HIV infection and the response to it. Since then, and for the next almost 20 years, not much happened in the construction of humanized mice. Progress was only slow and marginal until the mid-2000, when the NSG mice by Ito and then Schultz, Jacks were constructed. The NSG are not, which is a polygenic immunodeficient mouse lacking macrophages and so defective in cell function, etc., and defect and deficient in C5, and with sirpalonit receptor polymorphism. SCID, in that they are deficient in PK catalytic subunit, the DNA PK catalytic subunit, so they cannot recombine B cell variable gene or T cell variable gene, and deficient for the common receptor chain, the I2, the common chain of IL-2 receptor, 4, 9, 15, 17, and 21. So NSG have been used for the last almost 20 years very effectively, and they're sold by JAKs. They have some shortcoming. They have been grafted with human peripheral mononuclear cells or hematopoietic stem cells, CD34 cells, IV, IP, intrahepatically. The shortcomings are they require gamma radiation to myeloablate the mouse bone marrow to open up the niche for the human hematopoietic stem cells to settle and to differentiate and develop. And because of the gamma radiation, the myeloablation, they are prone to infections and wasting. Importantly, they show a limited immune response, particular antibody response, and they have a reduced lifespan. Some NSG mice have been made knock-in or transgenic for different human cytokines in an attempt to improve the immune response, particularly antibody response. But success has been moderate, best. So we decided, we attempted to construct a humanized mouse without this shortcoming by grafting human hematopoietic stem cells from cord blood, you see 34 stem cells, without gamma radiation. And we were very lucky, and luck is very important in life, in that there were two mutant mice, energy mutant mice, the NSGW41 by the Waskus group in Dresden, in Germany, and the MBSGW by Thompson groups in Madison, in Wisconsin. Those two mice are slightly different in background, genetic background. They have the same mutation, which is the KIT gene mutation, which encodes the c-KIT protein. The c-KIT protein binds cell factor and instrumental for developing stem cells, hematopoietic stem cells, in particular the bone marrow, to settle on stromal cells and then differentiate. As it happens, the mutation of mutated c-KIT makes it almost impossible for mouse hematopoietic stem cells to settle on the stroma and differentiate, therefore creating a huge advantage for the human hematopoietic stem cells. So our luck has been that in spite of the fact that these two papers were published in almost 10 years ago, they did not receive much attention by immunologists. So we use NSGW as well as NSGW41 mice to create, to construct humanized mice. And this is how we did it. We grafted MBSGW mice within 24, 48 hours of birth with fresh isolated human cord blood CD34 cells by intracardiac injection in an attempt to disseminate as much as possible the hematopoietic stem cells in all those site of differentiation, mainly bones, flat and big bones, and so maximize the chances of grafting. And then a big advancement was to condition these mice, this is at about 16 or 18 weeks, with E2, beta estradiol, estrogen. The rationale for that is both anecdotal and mechanistic. Anecdotal in that there are plenty of not very systematic studies showing that women mount 2, 3, 4, 5-fold better antibody response to viral and bacterial vaccines than men. It's very well known that investigators order from JAKs female biopsy or C57 when they're doing experiments with antibodies because they respond better. And finally, as you're well aware, 9 or 10 to 1 is the ratio of patients, female with lupus versus male. So that was the rationale for the estrogen condition. And here, we analyzed human CD45, which is a marker for all white cells, mononuclear cells, as well as granular cells. Over weeks after post-engraftment in those mice, that grafted mice and conditioned mice are referred to as THX, true human X. I'm not responsible for that. Our development office is. I think they were very impressed with X, such as SpaceX and Twitter became X. But I guess we can take X could stay for human xenograph. All right. So these are compared. And as you see here, in our mice, 95% or more, actually now more than that, are human CD45 cells, and this is the peripheral blood. And these are the Jackson mice, the NSG Jackson, bought from Jackson, and you see a peak about 50% of humanization around 15, 20 weeks. That then decreases dramatically. So there is a big difference in the grafting and in the maintenance of grafted and development, grafted human cells. And you see here the survival of the THS compared to CD34, NSG, Jackson mice. And these are 5 different THX mice. This is the spleen, and this is a site of analysis of 36 parameters. And indeed, THX mice shows human B cells, naive and memory plasma cells, early and late NK cells, all granulocytes, mainly neutrophils, as well as different T cells, CD4, CD8, central memory, effector T cells, etc. And again, you see this in these 5 THX mice. And in fact, the representation and the proportion of these cells, human cells, is virtually overlapping with what has been shown in 7 spleens from humans who died an accidental death, which is not shown. So we then, first of all, determine whether the B cell repertoire in THX mice is actually as diverse as humans. What you see here is the genomic representation of the 7 VH gene family, different colors, different family, denote different families, and gradation within a color denote the different members of the same family as you see here, the VH3, the largest, AV chain variable family in human is in red. And in 1, 2, and 3, this is the expressed B cell repertoire in 3 different human young adults. And you see that the expression is really stochastic and reflecting the proportionality and representation of those variable genes in the genome. The next 3 strips here, those are 3 different THX mice, and it's quite incredibly, similarity is quite incredibly tight with the expressed human B-cell repertoire and the genomic representation of those V genes. The same can be said about Jade's gene. Of course, they know the G gene is well known that in the human, the VDJ recombination often entails the usage of 3 different D genes chopped off and rejoined. Something similar can be seen in V kappa and J kappa, and V kappa, J kappa, light chains. And in fact, the heavy chain CDR3 length of these 3 THX mice overlaps with that of the 3 adult young humans here. And then we identify the different clone by sequencing the recombined VDJ sequence, right? And identify discrete clones. And now you see, as expected, and this is the mu repertoire, the putatively non-induced, if you wish, but in these 3 human subjects, you have a bunch of clones that are amplified, again, as expected, because these individuals were vaccinated, they had a flu in their life, and whatever else. In the EHX mice that are kept in clean conditions, there are fewer expanded B-cell clones and expanded to a lesser extent. Something very similar can be done, we did, and it's published in the T-cell repertoire. There is a complete overlap in the expressed T-cell repertoire. Now, if this is true, and it is, then if we take fresh isolated B cells, human B cells, from THX mice, and we stimulate it in vitro with classic genetic combination, plasma cell in differentiation of memory cell-like inducing stimuli, they should behave like fresh isolated B cells, human B cells from human, and they do. And you see here, in those B cells from THX mice, in response to a T-dependent stimulation, if you wish, CD154 plus cytokines, same stimulation on B cells of human B cells from humans. And you see here class switch B cells in this histogram, M, G, A, and E, humans memory B cells, so memory-like, and plasmablasts. Same exact things here, same in terms of germline and post-recombination transcripts, AID expression, and ICDA and PRDM expression. So, all is good, and indeed, when we injected these mice, THX mice, with this quintessentially T-dependent conjugated aptin, MP chicken gamma globulin, and analyzed the response to MP using MP5 in the solid phase, thereby capturing only high affinity antibodies. And we compared THX mice response, NGNE, to NAB-SG mice grafted with human CD34 cells, but not E2 conditioned here, as well as the CD34 Jackson NSG mice here. Now you can see that the THX mice mount a class-switched response, GNA and E2MP. There is some response in the grafted MBSGW, but not conditioned. Not much, and there is very little response in the NSG. Now, the response of the THX mice, in fact, entails the amplification of 3-4 major clones. You can see here in 4 different THX mice. And again, that's it's interesting to have these microclones that were intact, so to say, and then there is this selected clonal expansion. And I'm not sure that something like this has ever been shown in mice or humans, ever. And then we did the intraclonal analysis, and you see this beautiful intraclonal diversification in the major clones, selected clones. And the load of somatic mutations is actually quite reflective of what you will see in humans, in vivo. The same can be said, essentially, of the T-dependent antibody response, in this case to the MP-CpG, which is a TLR9 ligand, CpG, and this is the analysis of the response to the MP. And again, there is a good switch response G and A and E in THX mice involving somatic hypermutation and preferential clonal expansion in the gamma, or gamma, because now forget the equivalence between human gamma 1, 2, 3, and 4, and the mouse 1, 3, 2B, A, and 2B, that there is essentially no equivalence. So that's why we analyze gamma and intraclonal diversification here. Now, how can this happen? Well, it happens because THX mice show quite a nice spleen here, pretty much comparable to C57 in terms of size, but importantly, show what has never been quite found in humanized mice, well-developed lymph nodes in different districts over here. And this is compared to the Jackson mice and compared to the humanized MBSEW not hormonally conditioned, as you see here. So estrogen really does something. And then when we look at the spleen, then you see the spleen THX mice is structured. You see those germinal center that you don't see in the non-hormonally conditioned mice, and you don't see in the NSG. And then when you look at germinal center using the quintessential germinal center markers for B cells and T cells, CD20 and CD3, as well as the B cell, the KY67, BCL6 for central blast, AID, and BRIMP1, AID for somatic mutation residual combination, for plasma cell differentiation. Interesting, in the response to MPG gamma globally, there is a transition from a prevalent B1 phenotype in B cells to a B2 phenotype within 4 weeks. And germinal center, human germinal center B cells, and switch memory B cells can be identified in the spleen. Also, interesting, this can be the matter for discussion at the end of the presentation. What made it possible for THX mice to mount a high affinity T-dependent response is the presence of human thymus epithelial cells in those thymuses that are highlighted. They are marked in green here. You see very few or none in the non-conditioned grafted mice, but you see them here in THX mice. And those are the referred to also as thymus epithelial cells here. You see here 22 and 30 weeks after in those immunized mice. And in addition, in addition to TEX in the thymus, there are also human B cells here, which can also, together with TEX, can also contribute to the education, the development of T cells in the thymus. And what also can be found in the THX mice at a great proportion are T follicular helper cells, defined as 6LCR5 positive, PD1 positive human T follicular helper cells. You see them here, and they are not present essentially in NSG and virtually not in the non-hormonal conditioned mouse. And this is the quantification in disease struggle. Now, in the gut, and in this case, these are the DMP CpG immunized mice. You can see in the gut, in the gut you can see in the lamina propria, this human CD45 cell, B cells, CD3, you can see IgM, IgD, and IgA. In the spleen, switch memory B cells, plasmablast cells, plasmablasts, and plasma cells. And in the bronchoalveolar lavage fluid, valve, you can see again in the THX, but not in the non-conditioned human grafted mice down here. You see the something missing here. Those are, those are IgD, IgG, and IgA cross-switched cells. In the feces, we could find the IgD and IgA in THX mice, and in fact, we could find bacterial-bound in large numbers, bacterial-bound IgD and IgA. You see it's like 51, 52% versus 1% less in the non-conditioned mice. In addition, in THX mice, we could easily identify pair patches, and those pair patches contain T cells, CD4. They contain cross-switch B cells, switch memory B cells, plasmablast, and plasma cells. And these are quantified down here. And importantly, they contain sizable numbers of IgM positive, T positive, 27 positive, which identify marginal zone B cells in humans. That was very gratifying to show marginal zone B cells because, as you know, those are in the mouse have been shown to be important, or they are putatively important in bacterial infection as a first line of defense. Interesting, when we look at the microbiome in the gut, and then in these three groups, you have here THX mice, here the non-grafted MBSGW mice. So these are mice. These are THX mice. In the middle are the human grafted MBSGW, but not hormonal condition. So the non-grafted mouse have this predominance. Don't ask me to pronounce the name. I have underlined it here. Rickets bacillacea, the pink one, which disappeared totally in the THX, is still a little bit there in the transitional, grafted but not conditioned. And then in the THX, you have the green, which are the murid bacillacea. I'm not sure. The green, and more the yellow, and the blue. So it's very interesting that at the two extremes, you have the human that are really the heavy human, the green in particular. Green are actually particular of humans, not of mice. Here, you have those bacteria that are characteristic of mice. And in between, there is this transition. And you can see that nicely in the principal component analysis here. These are THX, these are the grafted non-conditioned, and these are the mice non-grafted and non-conditioned. So now, how about, given all this, can the mice mount a good antibody response to a vaccine? We use purified flagellin from Fragility fimurium to immunize THX mice and indeed induce a good cross-switch antibody response to DNA with B cell, IgG and IgA, B cell binding flagellin, generation of specific memory B cells, and plasmablast binding flagellin. And the serum from those vaccinated mice neutralize, as you see in a dose-dependent fashion, Salmonella typhimurium in vitro. These are your petri dishes, like we used to do many decades ago. And these are 5 THX mice sera compared for neutralization to 5 human adult sera, which are, of course, we all experience flagellated bacteria in our gut at the time. And so there is really no difference in the power of neutralization of the sera from the THX mice and the human. And here, these are the non-vaccinated THX mice here. Those responding B cells are mutated, good mutation, and with the select clonal expansion and intraclonal diversification, particularly in the alpha, which was respected to some extent in the response to flagellin. And those vaccinated mice are protected from a challenge with live, as shown here as compared to the non-vaccinated ones, with challenge with salmonella, and they don't die. And a similar successful vaccination response, neutralization, clonal expansion, intraclonal diversification, etc., generation-specific B cells are switched, binding, in this case, RBD. These are THX mice vaccinated with a Pfizer RNA vaccine, and the mounted class switch hypermutated neutralizing response to the RBD of the COVID-19 virus. The binding of mGNA is shown here, and this is the neutralization. This is the mutational analysis of those antibodies. And now again, this is very interesting. You see the select clonal expansion in the gamma, as expected, but even more so in alpha. And we found two reports, or three reports, on how this was unexpected. RNA vaccination, COVID-19 RNA vaccine in humans actually often induced a prevalent IgA response. And this response to RBD actually stays for quite a long time. These are IgGs, these are IgA. But you see, we analyze it up to 77 days. The IgG, the circulating IgG, are still there, quite sort of a constant level. And IgA go down a bit. So now both the flagellin vaccinated and the COVID-19 Pfizer vaccinated mice show good circulating level of human APRIL, human BAFF, human interferon gamma, TGF-beta1, IL-2, IL-4, IL-6, IL-10, IL-21. Well, there's no matter. IL-29 occurs in very low single-digit picogram. So I'm not sure that means much. But so obviously, these critical growth factors and cytokines critical for the mounting a good antibody response are there. And this may explain why all the other humanized mice don't respond, no detectable level of human cytokines of this sort with mice. And finally, THX mice can become a good model of lupus. So here you see, after one injection with pristane. Pristane has been used in some experiments to induce a lupus-like disease in mice. But in just one injection of pristane, you see these mice mount an antibody response to nuclear antigens, which is somatically hypermutated and entails a select B cell clonal expansion as well as T cell clonal expansion. And you see the deposition in the kidney. These are non-injected THX mice. These are lupus, what we call lupus THX mice. And you see here with the kidney damage and the mortality of those mice. And in fact, this is the last slide. And it goes back to why we attempted to generate humanized mice. In fact, the lupus THX mice with relatively advanced disease, with all sorts of anti-nuclear antibodies, initial kidney pathology, etc., and large spleen, can be reversed to a healthier state by administration of DMF. DMF fumarate, dimethyl fumarate, is actually used off-label, has been used off-label in a few lupus patients. It is used, the indication is the treatment or multiple sclerosis. And TMF, and this is a little complex, but take 1 minute. Fumarate it's an intermediate DCA cycle. It's an inhibitor of TAT2, catalytic DNA demethylator, which activates the ICDA and PRDM1 loci for AI-enabled implant expression and plays a primary role in the maturation of the antibody response. They fumarate inhibit alpha KG, which is the cofactor, one of the two cofactors, that do, the other one being ferrous to the and also inhibit GAP-PDH, therefore increasing the concentration of NID plus, which is the cofactor of SIRT1. And so this is another way in which fumarate dampens the overall response because SIRT1, we published this a couple of years ago. SIRT1 is an active deacetylator, an epigenetic factor. Deacetylate NF-kappa B, deacetylate histone H3K9, and deacetylate DMTNA methyltransferase 1. Deacetylate and DMT1 is active, whereas, of course, deacetylate NF-kappa B and histones are not. Deacetylate histone is associated with a closing chromatin, and deacetylate NF-kappa B is important because NF-kappa B is one of the two factors critical for induction of AICDA expression. And so this is how fumarate, and we know we are working on all this, this is how fumarate really could be an epigenetic therapeutic for lupus. There is more to it, and well, the more to it is close to our heart because we are. Is that TAT2 also recruit OGT for all Greek nicylation of histones, but as well as TAT2 itself. So, in addition to fumarate now, which is, so to say, a physiological inhibitor of the whole mature antibody response, class with some adaptation, hypermutated, and presence of abundance plasma stem cells and generation of B cells. So in addition to fumarate, we have mainly from Bristol-Myers Squibb small molecule inhibitors that can selectively inhibit the catalytic side of TAT2, as well as the OGT recruiting side. And so I mention all that because really this is why we started doing humanized science. And here are the people who did the work. These are the three people who did the work. Daniel Chap graduated almost 3 years ago now. Carlos Rivera, who is currently a postdoc, a senior postdoc in the lab. And Eli Zhu is currently a postdoc, but he will join Yale on March 1st. And Shiri Lee, who is the number one working on TAT2, and Griffith is a faculty in our department, and is the resident Thymus scientist in the department. And Karen Zhu, the some experiment, was a student in the lab as well. And we could not have done this, and we couldn't continue doing it without the help of the people in the OBGYN department, Patrick Ramsey, and importantly, Amy Bible and Belinda Lopez, who are incredibly generous with their time, and they call us as soon as they have the right puerpera with the right cord blood. Thank you very much.
If anybody else has questions too, there's another mic over there, or you can come get this one. But okay, we'll start with one question online, which is sort of related to a question I had, but asking, is the THX mouse model a good model for human leukemia, either PDX or cell line based leukemia? And then I was also curious about solid tumor, if you could use human cells.
Well, we have 3 groups that we are actually collaborating, but all of them doing solid tumors. I don't have an answer in terms of B-cell leukemia or T-cell leukemia. We haven't done anything, and we have not been approached by anyone wanting to do that.
And then another question, have you characterized also the myeloid cells in the THX mice?
Yeah, well, we show that the THX mice have human neutrophils, basophil, and eosinophil. We didn't do any. We didn't put any serious effort into the myeloid compartment. They seem to have all the elements of the myeloid compartment, but I don't know what. It's not our interest. That's the problem. Don't do anything.
More questions? Well, it's a really cool model, so thank you.
Thank you.
Okay, welcome back from break. A break that we initially thought was 30 minutes. It turned into 45. So the cowbell was a little early, but now we want to get started. It's, again, my pleasure and honor to be able to introduce another dear colleague of mine from UT Health San Antonio. Dr. Evelien Bunnik is an associate professor in the Department of Microbiology, Immunology, and Molecular Genetics. Evelien obtained her PhD from the University of Amsterdam and did a postdoctoral fellowship at University of California in Riverside before joining UT Health in San Antonio as an assistant professor. She's now built a really strong and well-funded research program, was recently promoted to associate professor with tenure, and is truly a rising star in her field. Her main research interest is to unravel the nature and acquisition of B-cell responses that protect against malaria in individuals exposed to Plasmodium falciparum infections. Her work has recently resulted in the publication of an important paper in Nature, and I actually mean Nature, not Nature Immunology or Nature Communications, Nature, demonstrating the presence of broadly neutralizing antibodies specific for the polymorphic erythrocyte membrane protein 1 adhesion proteins expressed by the parasite in severe malaria. Today, Evelien will tell us more about this exciting work in her talk entitled Towards a Vaccine Against Severe Malaria. Welcome to FlowTex 2025, Evelien.
Thank you so much, Mike, for that very kind introduction. And I'm going to dive right in and just show you a map of the world and explain that malaria is still a major global health, public health problem in lots of different parts of the world, mostly in sub-Saharan Africa. And if we look at the number of people who die from malaria, it's in this graph, it's color-coded by region of the world, and you can see that up to 95% or even more of people who die from malaria live in Africa. This graph also shows that sort of between 2000 and 2015, we've seen a steady decline in the number of people dying from this disease, mostly as a result of several different interventions, distribution of insecticide-treated bed nets, better access to healthcare, diagnosis and treatment, indoor residual spraying, so insecticide spraying to reduce the number of mosquitoes that can transmit the parasite. But from about 2015 onwards, there's been a plateau in this, in the number of cases, and in the era of COVID, we've even seen an increase due to the disruption of healthcare. And so we believe that we need better interventions to try and reach the goal of malaria elimination, to sort of get this curve to turn downwards again and ultimately reach zero. And one intervention that we think could be really important here is the development of effective malaria vaccines. And so the parasite has a very complex life cycle, and there are several different stages in this life cycle that can be targeted by vaccines. There are 2 vaccines that have currently been approved by WHO for use in Africa, and those both target the sporozoite stage, which is the stage of the parasite that is transmitted from the mosquito into the human host, into the skin. RTSS doesn't have very good efficacy. R21 is better, but there's still room for improvement here. And these vaccines really protect against infection. So they provide sterilizing immunity in people who are protected. But the problem is that if only one sporozoite escapes from these immune responses and travel to the liver, this can then cause malaria. Because in the liver stage, there is this massive amplification. One parasite can give rise to tens of thousands of daughter parasites that then seed a blood stage infection, and this is the stage where people get disease. And so escape from vaccine responses here can still induce severe disease. And so this blood stage then is a second vaccine target. The goal here is not to prevent infection, but to prevent disease, to be able to reduce parasitemia to such low levels that people don't get sick. And then there's a third stage that can be targeted by vaccines, which is the stage of the parasite that is transmitted back to a mosquito, and that in the mosquito undergoes development to make sporozoites that can then be transmitted to the next person. So these vaccines, the goal of these vaccines is not to, basically it doesn't have any benefit for the vaccinee, but will protect the next person. So at a population level, this can have a huge impact on malaria transmission. So today we're going to be talking about the blood stage, and specifically a vaccine, or sort of working towards a vaccine that can help prevent severe disease and can help prevent people from dying from malaria. And so I have to give you a little bit of background information here to understand what causes malaria. So one of the key factors, or one of the key mechanisms that underlie pathogenesis is parasite sequestration. So as parasites infect red blood cells, they export a lot of proteins into the cytoplasm of the red blood cell, and some of those proteins are expressed on the surface of the red blood cell. So these are parasite-encoded proteins exported and expressed on the surface of the red blood cell. And we call these Plasmodium falciparum erythrocyte membrane protein 1, or PMVP1 for short. These proteins allow the infected erythrocyte to adhere, so to bind to receptors on the endothelium and to adhere to the endothelium. And by doing this, they prevent clearance in the spleen. So it's a survival mechanism for the parasite. And if this happens in large numbers, there are several different receptors that these parasites can bind to. This leads to blockage of capillaries. So this mostly happens in the microvasculature and leads to complete blockage of the blood flow, but also elicits a response from the endothelium, including the release of pro-inflammatory cytokines, disruption of coagulation, leading to leakage of the endothelial barrier. And this is what underlies the most severe symptoms of malaria. And so just to show you, this is a capillary that's completely filled with parasites. So every black dot here in this capillary is a parasite. And so this is completely stagnant. There's no more blood flow because everything is blocked by infected erythrocytes. And especially when this process happens in the brain, this can have disastrous consequences. So these are, this is a a picture of a healthy brain. This is a brain of someone who died from cerebral malaria. And I hope you can appreciate here that this brain is very swollen. And the problem with brain swelling is that there's no place to go because we have a skull, and that really, there's no room for any kind of brain swelling without damage. And you can also see all these little hemorrhages in the white matter. Those are all just little blood vessels that have burst and cause lots and lots of damage. And so 80% of the people who die from malaria die because of cerebral malaria. And ultimately, this process, PNVP1 binding to a receptor on the endothelium, is what is underlying that mechanistically. So PNVP1 is a complex protein composed of multiple different domains. Every parasite has about 60 different PNVP1s, but every parasite only expresses one of those. And there is enormous diversity in the repertoire between parasites. So two parasites can have completely non-overlapping repertoires of PNVP1 proteins. The receptor binding ability of this protein is mostly determined by these first two domains, what we call the head structure. And I'm going to just separate those. They can be separated into three different phenotypes. There's one group that binds to EPCRs. Those are CIDR alpha 1 domains here. I'm mostly going to focus on the CIDR alpha domains. So CIDR alpha 1 domain can bind to EPCR. There can be a CIDR beta, gamma, or delta domain, which induces rosetting, which I'm just going to not explain for now because it's not the focus of my talk. And then CIDR alpha 2 through 6 binds to CD36. So these EPCR binders, they make up about 11% of all the PNVP1s that have been sequenced. So these are all the parasite genomes that have been sequenced, and about 11% of the PNVP1 proteins or genes that have been sequenced contain a CIDR alpha 1 domain. If you look in parasites isolated from people with uncomplicated malaria, it's about the same frequency, about 11, maybe a little bit more. Percent of the parasites have expressed a PNVP1 that contains a CIDR alpha 1 domain. If you look in parasites isolated from people with severe malaria, whether it's cerebral malaria or severe anemia or other forms of severe malaria, there's a massive overrepresentation of these CIDR alpha 1 domains. And so we believe, in part based on this data, but also other data, that parasites that express a PNVP1 with a CIDR alpha 1 domain play a major role in causing severe malaria. And this is, like, the process of parasite sequestration in itself is bad, but it's specifically binding to EPCR that has sort of an added layer of negative effects. So EPCR is endothelial protein C receptor, and it's a really, really important receptor for maintaining endothelial homeostasis. So if the parasite binds to EPCR, it can no longer perform its normal function. And its normal function is to regulate anticoagulation and also to maintain an anti-inflammatory state of the endothelium. And so binding of PNVP1 infected erythrocytes to EPCR disrupts coagulation, and it disrupts the barrier function of endothelium. And so preventing this interaction between CIDR alpha 1 and EPCR will, first of all, decrease cytoadherence. So it will keep parasites in the circulation and make sure that they're removed by the spleen. It will increase the endothelial barrier function, reduce inflammation, and all of that together will limit pathogenesis and prevent severe malaria. And so it's long been recognized that CIDR alpha 1 is a good vaccine target. The problem is it's highly diverse in sequence. So this is a phylogenetic tree that shows you sequences of different CIDR alpha 1 domains from different parasites. And there is a lot of sequence diversity, so much so that these CIDR alpha 1s themselves can be classified into different subclasses just based on their sequence. That's also illustrated here. This is a sequence logo, so sort of an average of 800 different CIDR alpha 1 domain sequences. And you can see some residues are conserved, specifically the cysteines are conserved because they're important for folding of the protein. And a couple other amino acid residues are conserved, but overall there is just immense heterogeneity in these proteins. And so this is not a good vaccine target, really. I mean, it's a very difficult vaccine target. It's good biologically, but it's difficult from the perspective of making a vaccine and eliciting immune responses. And this is just to illustrate that point, this is the data from a preclinical vaccine trial where animals were immunized with a CIDR alpha-1 variant that was based on the CIDR alpha 1.4 domain. And you can see that it's immunogenic. It elicits immune responses or antibody responses, but only against CIDR alpha 1.4 variants, not against 1.1s, 1.5, 6, 7, or 8, right? So it's subclass specific, and there's no breadth in these immune responses. And so one possibility would be to have a multivalent vaccine where every subclass gets its own immunogen, but that's not ideal. And the reason we believe that that's not strictly necessary is because if you look at people who live in malaria endemic regions, severe malaria is something that really only happens in very young children. So this graph shows you age on the x-axis, and on the y-axis it's the basically percentage of people that are susceptible to severe malaria in red. And you see this line come up sort of after children have stopped drinking mother milk, so getting maternal antibodies, they become very susceptible to severe malaria, but only for a very short period of time. So as soon as they've had 2 or 3 or maybe 4 infections, they develop an immune response that protects them against severe malaria. And this immune response, one of the first antibody responses that comes up in these very young children are antibodies against the EPCR binding CIDR alpha-1 variants, shown in blue here. And so we know that our immune system is able to make good antibody responses against these P and VP1. So all we need to do is understand how the immune system does that and then mimic that by vaccination. So if we take plasma samples from people who live in malaria endemic regions, we can purify antibodies against these CIDR alpha-1 domains. And so if we do that for, say, we use a CIDR alpha 1.4 variant to purify all the antibodies that bind to that particular variant, we actually see reactivity across the board against all of these different variants. And so what I'm showing you here in blue is reactivity, so binding of antibodies against all of these different CIDR alpha-1 variants. And in red is inhibition of EPCR binding. So the darker red means that there's less or more inhibition of EPCR binding. And so if you take antibodies that we know bind to 1.4, we also see antibodies, or some of those antibodies can also bind to 1.1s or to the 1.6 or 1.7 or 1.8 variants. And so this affinity purified serum IgG has broad reactivity, and it can block the interaction between CIDR alpha-1 and EPCR. But we don't know exactly how broad those antibodies are, right? It's impossible to tell because this is still a polyclonal collection of antibodies. And so we don't know what each individual antibody recognizes. And we also don't know exactly how those antibodies interact with CIDR alpha-1. And so that's where the project that I'm going to tell you about basically started with. This was the data we had and the questions we had. And so the approach we took was to look at B cells and clone monoclonal antibodies, because with monoclonal antibodies, we know exactly what they recognize, and we can tell, we can sort of interrogate how they interact with CIDR alpha-1. And so I'm going to show you basically our pipeline, or the workflow that we followed. We started with samples from people living in Uganda, people who have lifelong malaria exposure. We isolated antibodies with B cells with specificity to CIDR alpha-1, cloned out their monoclonal antibodies and expressed those recombinantly, and tested those antibodies for recognition of different CIDR alpha-1 variants. measured how well those antibodies were able to inhibit the interaction between cyto-alpha-1 and EPCR, or in a more advanced assay between parasite-infected erythrocytes and endothelium. And then finally, we performed structural biology to understand the interaction at a molecular level between the antibody and its antigen. And this was a large collaboration, so I just want to put a few faces of my collaborators in this picture as well and give them credit for their work. We work with Brian Greenhouse at UCSF, who runs a cohort in Uganda, has been very generous in sharing samples from that cohort with us. Rafael Reyes was a PhD student in my lab who performed the isolation of cyto-alpha-1 specific B cells and expression of the monoclonal antibodies in some of the early screening. We collaborated with Thomas Lefsen at University of Copenhagen in Denmark, who has shared proteins with us to perform the cyto-alpha-1 specific B cell isolations, and also did some of the downstream screening. Maria Bernabeu at EML Barcelona performed inhibition assays to test how well those antibodies are able to inhibit parasite sequestration. And then finally, there were two structural biology labs that helped us doing X-ray crystallography and cryo-EM of our antibodies in complex with cyto-alpha-1. All right, and so because this is a flow cytometry conference, I thought normally I would skip over this, but I thought I'll just put in my gating strategy of how we isolate antigen-specific memory B cells, or antigen-specific B cells, really. So we start by performing a negative selection on B cells. So we start with roughly, purified B cells, not completely pure, but enriched for B cells, and then gate on CD19, and take the IgM negative, IgA negative population, which effectively gives us an IgG positive population, but we didn't want to select on IgG positive because it interferes with some of the antigen tetramers. So after this step, we perform gating on our antigen tetramers. And we make those ourselves using a fluorochrome conjugated streptavidin that we couple to biotinylated proteins. We produce the biotinylated proteins, in this case, cyto-alpha-1 proteins ourselves, and then mix them with our streptavidins to make an antigen tetramer. And now there's two different approaches to isolate or gate on antigen-specific B cells. You can either make two tetramers in two different fluorochrome formats and gate on the double positive cells, or you can make a decoy tetramer, an antigen tetramer and a decoy tetramer, where the decoy tetramer is the same streptavidin, but now labeled to a second fluorochrome and coupled to a non-specific or a non-relevant protein. And so in this strategy, all the non-specific B cells, or all the B cells that bind to your tetramer, to any part of your tetramer that's not your antigen, will also bind to your decoy. And because there's two colors here, they will form this, they will be sort of located on this diagonal. And so your antigen-specific population of B cells is now single positive for BV421 and showing up as a small population here. All right, and so this is what Rafael did in my lab. He made tetramers for two different cyto-alpha-1 variants, a 1.1 variant and a 1.4, sorted antigen-specific B cells and isolated monoclonal antibodies. And this gave him a couple different sort of monoclonals, different classes of monoclonal antibodies. There were some that were subclass specific. So he had one antibody that recognized only 1.4 variants and one that only recognized 1.1 variants. And there were a couple monoclonals that recognized 2 or 3 different subclasses. So they were broader, but not super broad. And then there were these 3 antibodies that we were very excited about, because they recognized 5 out of the 6 subclasses. So essentially every single variant from 1.4 through 1.8 was recognized and inhibited by these 3 antibodies. So C7 and C62 turned out to come from the same donor and were clones of each other. And so we decided to continue with only one of them because they're essentially the same. So we went forward with C7 and C74. And the next question we wanted to answer, just to confirm, because all of these experiments, this screening was done with recombinant proteins, we wanted to make sure that these antibodies can bind to PNVP1 on infected erythrocytes. And for this, we also used a flow cytometry-based assay where we gate on, so we take parasite culture, we enrich for parasite-infected erythrocytes, gate on erythrocytes, and then are able to separate the infected red blood cells that contain parasite DNA from the uninfected parasites, the uninfected erythrocytes using a DNA stain. And because even though we enrich these parasite cultures for parasites that express a particular variant of PNVP1, there's always some low-level switching, and so part of the population has switched to expressing a different PNVP1 variant. And so we stain for expression of our particular PNVP1 variant, but there will always be a population, like a subset of parasites that will not express our PNVP1 of interest. And so these are our controls. So you can see that we have about 85% of our erythrocytes is infected. There's a small uninfected population. And there is a good, like the majority of the parasites expresses the PNVP1 that we're interested in. But again, there's a small population of parasites that has a different PNVP1 on its surface. And so this is then staining with C7 and C74. And the quantification of those data, we've done this for 3 different parasite lines. So it's either alpha 1.1 that's not bound by C7 or C74, and then a 1.4 and a 1.6, where we see, in both cases, we see binding for both monoclonal antibodies. And so they bind to PNVP1 in its native form on infected erythrocytes. So that's good news. And now, as a next step, we were very interested to see if those antibodies can actually inhibit sequestration of the parasite. So binding, so attachment of the parasite or infected erythrocyte to the endothelium. And to answer this question, we collaborated with Maria Bernabeu in Barcelona, who has this really cool 3D engineered microvessel assay where she can basically make, in vitro make capillaries and flow blood through those capillaries and measure parasite sequestration. So she creates these little microfluidic networks in collagen that she can seed with primary human brain endothelial cells that form a nice monolayer around the network, and then perfuse this network with parasite-infected erythrocytes. And you can see, so the large blue blobs here are the nuclei of the endothelial cells. The smaller, brighter specks in the bottom are parasite-infected erythrocytes that are attached to the endothelium. And because this is a network with an inlet and an outlet, the flow rate or the flow velocity throughout this network is not the same everywhere. It's higher in the corners and then, or higher in closer to the outlet and the inlet and lower in the corners. And so she measures parasite sequestration across different flow velocities or across different wall shear stresses. And that's what I'm showing you here. So on the x-axis is wall shear stress. On the y-axis is sequestration. And in gray is our control, where you see that there's more sequestration at lower flow velocity or lower wall shear stress rates. But at all wall shear stress rates, you see that there's inhibition of binding when you perfuse in our antibodies, C7 and C74. And again, we did this for 2 different parasite lines expressing different CIDR alpha 1 variants. All right, so far I've shown you that we've isolated monoclonal antibodies with reactivity against 5 out of the 6 PNVP1 CIDR alpha 1 subclasses, and these antibodies inhibit parasite sequestration under physiologically relevant flow conditions. And so we were very interested in learning how C7 and C74, well, where they bind and how they bind, how they deal with the sequence diversity between these different CIDR alpha 1 variants. And so we did structural biology. So just to sort of to orient yourself, if we take a CIDR alpha 1 variant, then there are two alpha helices that together make up the EPCR binding site. And from X-ray crystals that were, or structures that were generated about 10 years ago through X-ray crystallography, we know that those two alpha helices are sort of located here and interact with EPCR. And there's this phenylalanine residue in particular, and I'll come back to that, that sort of sticks out and is captured by EPCR. It kind of sits in a binding pocket formed by EPCR. Those same two alpha helices are bound by C7. So this is X-ray crystallography data that shows how C7 interacts with CIDR alpha 1.4. And if you overlay this structure with EPCR, the old structure of EPCR in complex with CIDR alpha 1.4, you see that they're sitting right on top of each other, right? So there's this direct blocking of EPCR binding by antibody C7. So this is for a 1.4 variant. We've done the same for a 1.7 variant, and essentially there's the exact same orientation. Interaction is exactly the same. And we've also done it for a 1.6 variant for C74. And again, it's binding in the exact same way. And just to illustrate how different in sequence those different variants are, I shown here in red the only residues that are identical between the different variants, right? There is sequence variation at every single amino acid position. All right, and so these two antibodies bind the EPCR binding site directly, and in that way block EPCR binding. So what about 1.1? What's different about 1.1, right? Because they have no reactivity whatsoever against the 1.1 variants. So what I'm showing you here are the EPCR binding helix and EPCR binding supporting helix for the 1.1 through 1.8 variants. And in the top and then in the bottom is are the 1.1 variants. I've shown here, if circled in yellow, the contact residues that are shared between C7 and C74. There are only 4 residues that are conserved among all of these different variants. And of those 4 variants, 2 are also conserved in 1.1s. So those are not the ones that make the difference. So there's only 2 that are different in the 1.1. So this phenylalanine is a leucine in 1.1s, and this I'm blanking on the name now. This E is actually shifted 1 position upwards in the 1.1 variants. And so what we did first is make mutants in the background of a 1.4 variant to show that those residues at F, these 2 Fs and this E, are essential for binding of C7 and C74. So if you mutate this first phenylalanine to an alanine, you lose binding. Not everything, but there's a significant drop in binding. The second phenylalanine, this is the phenylalanine that's sticking into that EPCR binding pocket. It's also absolutely essential for binding C7 and C74. And then finally, this E here is also, you also see a large drop in binding if you mutate that to an alanine. Now, if you start with a 1.1 background, there's no binding by C7 or C74. If you sort of do the reverse now, if you change this leucine to an phenylalanine, there is maybe a little bit of binding for C7, but nothing to write home about. If you change the EKG here to the context in the 1.4 through 1.8 variant, so EA, there is again a little bit of an increase for C7, but nothing much for C74. But if you introduce both of those changes at the same time in the 1.1 background, now you see that there's really, really strong binding of C7 and C74. So those residues are basically all that these antibodies need to recognize the Cyder alpha variant. Doesn't matter if the rest of the protein is more like a 1.1 variant or more like a 1.4 variant. If those residues are mutated away, you lose binding. If you introduce those residues into a different background, you have binding. And so that's really cool, and it makes us understand, at least to some extent, the reactivity of these antibodies. It still leaves us with a problem because we still, okay, let me first come back to my conclusion. So the binding is dependent on these 3 highly conserved amino acid residues that are different in the 1.1 variants. And again, so this leaves us with a problem because ultimately, if you want to make a vaccine, we want to have reactivity against all of the 6 different subclasses, not just 1.4 through 1.8. And so one thing that we're really excited about or really interested in and going to put effort into is to try and find antibodies that inhibit all of these different Cyder alpha 1 variants, because EPCR, like all of these variants bind to EPCR. So if we can find an antibody that even more closely mimics the way that EPCR binds to Cyder alpha 1, I think there is potential for finding antibodies that are pan-reactive against all Cyder alpha 1 variants. We have recently isolated a new antibody that has reactivity to all of the 1.1 variants and some of the 1.4 variants as well. So far there was this clear dichotomy, like either if you have reactivity to one, you don't have reactivity to the other. But this antibody, to some extent, bridges that gap a little bit, and so we're very interested. I've heard the Sai, who is doing the cryo-EM for us, he has a structure. He's still working on refining it, so I can share it with you. But I'm very curious to hear in the coming weeks sort of what his insight is on how this antibody is able to bridge and bind to both 1.1 and 1.4 variants. This also gives us a cocktail of 2 monoclonals that we could potentially use as prophylaxis. So prophylactic immunization has been tested in malaria for antibodies against the sporozoite variants, the sporozoite stage that is transmitted by mosquitoes into humans and has been very effective. And so possibly this is a cocktail of antibodies that we could test to see if this strategy works and can prevent severe malaria ahead of making a vaccine. But we are also interested in designing a vaccine and designing immunogens now that we know how our C7 and C74 antibodies interact with these variants. And so we've done some preliminary work in collaboration with Neil King at the University of Washington, who has developed this AI-based method to design proteins based on structures. And so he designed, out of his pipeline came about 95 different variants that were predicted to bind to C7 and C74. After expressing and purifying them, we were left with 85 different variants. We then screened for binding, and 6 of them have high affinity to C7 and C74 at sort of similar or higher affinity than the original Cyder alpha 1 variants. And so these are the 6 different variants that came out of that pipeline. They have the 2 alpha helices that are similar to what's present on Cyder alpha 1, but they have a very minimal scaffold, and they can be sort of grafted onto like a virus-like particle or something like that to have like highly repetitive exposure to the immune system to elicit strong antibodies. And so we're excited about testing these different immunogens and seeing what kind of antibody responses we get out of this. All right, that's what I had for you. I learned this morning that if I show you cat pictures, I don't get hard questions. So these are some of my cats. And with that, I'd like to thank all the people who contributed to this work. It was really a massive effort from a lot of different labs. And I'm happy to take any questions. Thank you.
So those cross-neutralizing antibodies or cross-reactive antibodies, 5 and not 7, 7 and 7.4, 5 and 7, whatever. So formally, they are not polyreactive antibodies. They are antibodies that recognize the same residues in different contexts,
Correct.
as shown by your 1.1,
Yes.
which when you put in the right amino acids, right, in the context is different, but then the antibodies react.
Yes, correct.
So that's, am I saying it right?
Yeah,
Is
yeah,
that
yeah.
right?
That was right. Yeah.
So then to have an idea of how effective those antibodies, or in general, antibodies of that kind can be in vivo. What is the KD or K-on range of those antibodies if you do an analysis using relatively big peptides, right? Just partial, not the whole structure of the that is involved in the recognition by the parasite of the to the
So
endothelium.
we've tested the binding affinity to just the whole cider alpha 1 domain, and the affinity is in the low nanomolar range. For some of the, it's like high picomolar for some of the variants, low nanomolar for others. So it's they're pretty high affinity.
Meaning 10 to the, in terms of KD, are you talking KD or
So you just mean binding, like association rates?
K-on? Or
Okay, yeah. So binding affinity is calculated by the combination of K on and K off.
yeah,
I don't remember what the K on and K off values in the individual values were, but together they have a very high. They give you a very, so they go on and then they basically stay on. They don't come off a few.
and the KD, have you any idea what the KD is?
I would have to look it up. I have the data for you, but I have to look it up.
No, I say that because given the fact that what you want is to have quite a persistent binding on the antibody, right?
Yes.
To the parasite, to inhibit the binding to the endothelial cells. So the KD can be very important.
Yes. Well, I mean, that's what we see. We see that the antibodies bind very strongly and then they don't really fall off. So if you have your binding curve
KD is
going
good.
up and then they're basically flat binding.
So now another point that is speculative, more is so is there any room, or do you think, for polyreactive antibodies, meaning antibodies that recognize different residues in similar contexts or different contexts? Because when you think about the HIV story, it has never been clear to me whether these people are looking at polyreactive antibodies or antibodies more like yours that recognize the same epitope in different contexts.
Yeah, I mean,
so specific antibodies.
These antibodies to me seem very specific.
Yeah.
Because like you said, basically proven by the point that they don't recognize the 1.1s, even though they're very similar in structure, but just miss one or two key binding residues. So I'm not sure. I'm not sure what polyreactivity has to do with this. And I know the story from HIV that you have.
Yeah.
Some of
I'm just asking whether you think that there is room for polyreactivity. I mean, whether it's almost a belief that you can have an effective cross.
Yeah, it's an interesting question. There's actually, so there was a paper published in Immunity last year that showed that if you isolate sort of the autoantibody fraction from plasma from people who live in malaria endemic regions, that it has anti-parasite activity. So it binds to parasite proteins and it can inhibit parasite invasion. But yes, but there's cross-reactivity to human proteins. And I don't know what to do with that. Like, I don't know what that means. If it's just true, if it's cross-reactivity or
So,
yeah.
so you're, just to clarify, you're asking if it would be beneficial to generate a lower affinity antibody that has a higher propensity for cross-reaction that could be protective. Yeah, rather than these highly specific high affinity antibodies. Yeah.
Never quite do. While HIV people
Yeah.
want what they want, cross-neutralizing antibody.
Yeah.
That's the
I'm not
first
sure if that's necessarily what they want, but I think that's just something they observed.
thing. Well,
But and to some extent, it like for some of the epitopes that are close to like lipid membrane, lipid bilayers, yeah.
And we did find, we did find IgG that were only reactive, yet they had a good IgG for insulin. So I don't have an answer. I'm not even sure that we did have, I'm not even sure that something that is only from as to necessarily be not very fascinating. When one point one, we have those two, we have two.
Mm-hmm. Yeah.
The reality is true that we are used to the you know, you think about it, the epitope, 4 or 5 amino acids.
Let's hope it is.
Okay, thank you all for still hanging around here. It's my great pleasure and honor actually to introduce my previous mentor, because 3 years ago she introduced me for my PhD defense. So this is very exciting. Mitty, her full name is Amitinder Kaur, frequently goes by Mitty. And she not only runs her own lab, as if that wasn't enough, she also runs a name of a lab that's really long. I always have to read it. It is the Non-Human Primate Cellular Immunology Core Laboratory. And she's taught me to be a great scientist because she is a great scientist. Not only that, she's also a great mentor. And I hope you can see in the 2 stories she's going to share today about how she's a great scientist and it's focused on details that people sometimes forget about. She wanted me to give a very informal intro, so I'm doing that. And her passion for helping patients in India is what drove her to doing preclinical research. And that drove her to the US, where she worked on the natural hosts of SIV to work on HIV, preclinical correlates protection. And that, through some serendipity, drove her to work on congenital CMV, which she's going to talk about as well. And I hope you guys find these two stories interesting. So take it away.
Thank you, Matilda. That was quite embarrassing, but I really appreciate this invitation to talk here today. And, you know, just following the elegant talk just now, this is not going to be like that, but it's going to be different. No cats, but I'll show you monkeys. So I'm from the Tulane National Primate Research Center, which is really, you know, one of the best primate centers in terms of the rhesus macaque breeding colony that we have, and that the studies that are being conducted there. So my lab has basically had sort of 3 areas of research interest in CMV, AIDS, and basic immunology, T-cell immunology on natural killer T-cells. And today I was going to give you 2 stories, one about some of my past work on natural killer T-cells, and then some of the more recent work on congenital CMV. So the way I look at non-human primate models of infectious diseases is really, I've been interested in pathogenesis studies to understand the immune basis of protection, and that those pathogenesis studies to then inform vaccine design. So the first part of my talk is going to be on SIV infection, that is the monkey analog of HIV infection. And this work has really focused on natural and non-natural hosts of SIV, which I'll explain more. And that led me into looking in natural killer T cells. And then the second part of my talk will be on rhesus CMV infection. And this is all going to be like a little, not so much detail, but a little high-level view. So if we look at the primate evolutionary tree, so there are like 3 groups of primates. The hominids is the humans and great apes, and then we have the old world monkeys, and then the new world monkeys. And we're all supposed to have like a common primate progenitor, about 60 to 80 million years away. This is really sort of, this tree is important thinking about SIV infection. So a lot of really seminal work has gone on to show that in Africa, so in the old world monkeys, you have the Asian monkeys, which are the macaque species. And then in Africa, a bunch of non-human primate species, which before I came to the primate center, I never knew existed. But there's a plethora of really African non-human primate species. And investigators have looked into many of these species. And to date, there were at least 40 of these non-human primate species that have, from which their own SIV has been isolated, which shows that these species-specific SIVs have co-evolved with their host, so they're really ancient. And the ones that we know most about are the SIV chimpanzee, which we now know that HIV-1 originated as a cross-species transmission to humans. When it went into gorillas, you got the SIV gorilla. And SIV chimpanzee itself is actually a recombination from SIV in the great spot-nosed great spot-nosed Mona monkey, and then the red-capped sooty mangabe. Now, for HIV-2, we know that SIV in sooty mangabes, that when it crossed into humans, created HIV-2. Now, these cross-species transmissions, for the most part, are abortive. So the SIV in natural hosts, as far as we know, for the most part, are asymptomatic in their natural hosts. But when they cross into a different species, they could produce AIDS. Several years ago, like when HIV first became known, then both at the New England Primate Research Center and at the California Primate Center, they started seeing macaques that had an immunodeficiency-like syndrome from which SIV MAC was isolated. And this SIV MAC now, we know, was actually a cross-species transmission from SIV and pseudomangabes that were at the Tulane Primate Center. That was called the Delta Primate Center at that time. And pseudomangabes were a host for leprosy, lepromatous leprosy. So there were studies that were done that were injecting lepromatous nodules into rhesus macaques to induce neuritic leprosy. And sort of the original founder of this SIV MAC was one of these monkeys that had been shipped to California, then to New England. So it's actually like a fascinating forensic history. So anyway, most of the experimental work that has been done in natural hosts has been SIV in pseudomangabes and then in African green monkeys. So when I started looking at SIV infection in natural hosts, the most sort of important finding, the question was, why don't they develop AIDS? And so when you look at the non-natural host, which could be HIV in humans or SIV in rhesus macaques, the typical causes that you have, like acute viremia, which then settles down to a set point, and then over time, they progress to AIDS, the plasma starts, SIV starts rising. And during this time, you have an initial dip in the blood CD4 counts, which recovers, but then gradually declines. You have a massive acute decline in gut CD4 T cells, which remains persistent. And then you have this activation, immune activation of T cells, which subsides, but then remains at above normal levels. And that's been called this chronic immune activation. So when I started looking in pseudomangabes, at that time, there was a cohort of SIV negative pseudomangabes at the Yerkes Primate Center in Atlanta that we could infect. And basically the findings were that when you infect them, they develop an acute viremia and they have a persistent set point viral load, which in a non-natural host will result in AIDS, but it doesn't result in AIDS in the natural host. So looking at what happens to the CD4 T cells, it's the same pattern as in the non-natural hosts, that you have both blood and gut CD4 T cell depletion. The difference is that the gut CD4 T cells start recovering slowly, but never to their normal levels. And you see this immune activation in acute infection, but it subsides. So studies from my lab and several other labs have shown this phenomena in pseudomangabes and in African green monkeys. And really sort of the holy grail has been that what are the mechanisms underlying early resolution of immune activation? Because if we could figure that out, that may be key to protection against AIDS and really important for a vaccine which is not a sterile, which does not induce sterile protection, but could still help to prevent AIDS. And at that time, I was interested in cellular immunology, T cells. So one of the first things I looked and asked is that because they don't have this chronic immune activation, maybe and they have these persistent viral loads, are they tolerant? Don't they mount an anti-SIV T cell response? And in fact, they do. So that's what got me into innate immunity, because thinking about it, I felt that the early events is really a determinant of the course of immune activation. And so figuring out what is different very early on may hold the key. And because my interest has always been on T cells for innate immunity, I went to T cells. So among T cells, it was these natural killer T cells and other innate T cells. The questions I was asking is, do they modulate immune activation? Could they shape adaptive immunity and alter the course of SIV infection? And so these were studies in 2010, published in 2010, 2012, which in pseudomangabes, first characterizing the natural killer T cells, and I'll describe some of these. So looking back a little bit about what are unconventional innate T cells, so they're really a class of T cells, which I think may be, again, growing in the different types of these T cells. But their characteristics are that they're not restricted by classical MHC, class 1 or class 2 molecules. And they are innate. A clue to their innateness is that whatever they recognize on the antigen-presenting cell, it is functionally monomorphic, which is not a feature of adaptive immunity. And most of them recognize non-peptide immunogens, and they are sort of preserved across species. So you have a similar recognition pattern within individuals of species. And in terms of just schematically thinking of when they come, they probably are important in the soon after infection, whereas your conventional MHC restricted T cells come on later and are specific. So among the natural killer T cells, there are two types of NKT cells, type 1 and type 2. So both type 1 and type 2 NKT cells, by definition, they are CD1D restricted. And CD1D is a non-polymorphic MHC class 1-like molecule. So the type 1 NKT cells is what I will be talking about. They are called invariant NKT cells, and they were actually first identified in mice. And the reason they were called NKT cells is because they were T cells that express the NK marker, NK1.1. And when the T cell receptor was sequenced, it was found that it was the alpha chain, the CDR3 region of the TCR alpha chain was invariant, and then the V beta chain was restricted. In humans, the equivalent iNKT TCR it's the V alpha 24, J alpha 18, which is paired with V beta 11. The type 2 or variant NKT are much less studied because they're really harder to study. But there is a group that have defined type 2 variant NKT cells as recognizing sulfatides. But they're all, I think that the hallmark of an NKT cell is that it's CD1D restricted. And characteristically, they are functionally, the same cell can produce both Th1 and Th2 cytokines. And they recognize glycolipids presented on CD1D molecules. And the type 1, the invariant NKT cells, the classical glycolipid is alpha-galactosylceramide. So just to sort of visualize, like when you look at a glycosphingolipid, say in this case, the alpha-galactosylceramide, there's a sugar moiety, there's a fatty acid chain, and how, beautiful studies on crystal structure have shown how, the different binding of, a conventional T cell, which presented on, say, a CD8 T cell on an MHC class 1 molecule, it's really a 9 to 11 amino acid, which, there are pockets for the second and the 9th amino acid chain. Whereas in a CD1D molecule has a sort of a deep hydrophobic groove in which the fatty acid sits, and then it's the sugar moiety which connects with the CDR3 region of the alpha chain. And so sort of the whole thing is sort of like rotated. So just differences between invariant natural killer T cells and classical T cells. The T cell receptor is invariant as opposed to in classical T cells being really polymorphic. That is a feature of adaptive immunity, so that a T cell receptor is only against a particular defined peptide specificity and class 1 restriction. The antigen is lipids for natural killer T cells. It's peptides for your classical T cells. And then, the time to effector function is very rapid. And so the type of immunity is innate as opposed to adaptive. So thinking about it, how do these iNKTs get activated? Because if what they recognize is lipids presented on CD1D molecules, there are very few microbes which have these. And the classical ones are Sphingomonas, Borrelia. So if you get infected by one of these pathogens, your antigen-presenting cells could upregulate CD1D, present these microbial antigens, and the iNKT recognize them. But really, iNKTs, can be activated in a variety of different situations. So there are two other models of how can they get activated. One is that whenever you get infected or you see a danger signal, those toll-like receptor mediated activation upregulates endogenous self-lipid loaded CD1D molecules that the iNKT doesn't care what is being presented to it, as long as it's a lipid and a CD1D molecule. And then the other way is TCR independent activation, where the iNKTs express IL-12, IL-18, and IL-15 receptor, as do other innate T cells also, so that dendritic cells secreting any of these cytokines can activate iNKTs. So in terms of what they do, they are early responders, and just like every part of the innate immune system, NKT cells act as an interface between innate and adaptive immunity. So they can have direct effector function, that activated iNKTs can be cytolytic and produce the different cytokines. But they also have a major part of how they act is through their downstream effects on other cells. And so iNKTs, when they get activated, they produce interferon gamma, they upregulate CD40 ligands, so you have enhanced DC maturation, and that can result in better T cell responses being induced. They also activate your NK cells and then your B cells. They also interact with Tregs for their immunoregulatory function. And mainly murine studies, a bunch of them showing that iNKTs are implicated. They have an immunoregulatory role in suppressing graft tolerance, suppressing type 1 diabetes in mice, and then effector function mainly in tumors and pro-inflammatory in asthma. So they're really pleomorphic. So when I started looking at iNKTs, the question was, how do I start trying to identify them? It's a rare population, and in humans, as well as in monkeys, there's no identified analog of the NK1.1 receptor. So if there was, that would be very easy. People have implicated CD161. That is not the case. A lot of studies that are published, even in high-profile publications, they call iNKTs as CD3 positive, 56 positive, but these are not iNKTs. When you look at the golden standard of iNKTs, it is really, you have to show a T cell which will be able to bind to a CD1D molecule presenting a glycolipid, in this case, an alpha-galactosylceramide, an analog. And so the NIH Tetramer Core facility has these unloaded CD1D tetramers, which are empty, and then they also have this PBS 57 loaded CD1D tetramers. And PBS 57 is just alpha-galactosylceramide. I think PBS is Paul Savage, I think the person who made these. And so that's why. So this is really the golden standard. And when you look at iNKTs defined by this golden standard, most of them are 56 negative. If you gate on all the 56 positive T cells, most of them are not tetramer positive. But I still find these publications, and so whenever I get the bully pulpit, I have to say this. So the other way that you could identify NKT cells is so the 6p11 antibody was actually made against the invariant CDR3 portion of the V alpha chain. So the 6p11 antibody binds specifically to the type 1 iNKTs. And in humans, a lot of you can use dual staining with the anti-V alpha 24 and the anti-V beta 11. But in monkeys, we can't do that. The V alpha 24 antibody is cross-reactive, but not the V beta 11 antibody. So in the first studies, how we started looking, we used the golden standard of tetramers as well as 6p11 staining to identify iNKTs. And this is just a schematic showing that on a tight lymphocyte gate, it was important while gating on CD3 cells to really do this funny type of gate where, because a lot of the iNKTs, they are activated, so they downregulate their CD3. And so if you just do like a rectangular thing, sometimes you miss your iNKTs. So we have this kind of gating to gate the CD3 positive T cells. And just to note that in monkeys, the TCR, alpha beta TCR antibody doesn't cross-react. So CD3 is really taken as a marker for T cells in monkeys. So on the T lymphocytes, then just gating V alpha 24 against the tetramer and showing that you see a nice population, but with the unloaded tetramer, you don't see anything. So initially, I screened 50 pseudomangabes and with both the unloaded and the loaded tetramer. And basically could show, because these are rare populations, we had to collect at least, like we stained with 3 to 4 million PBMCs per tube, and then made sure that we were collecting at least 200,000 CD3 positive events, because it's only then that you had a sufficient number of events to have confidence that this was not noise, but this was a true signal. So based on that, except for one animal which had like more than 0.02% of with the unloaded tetramer, otherwise in all the other animals, we sort of did a cutoff of 0.02% as being positive. And with that, roughly half of those 50 pseudomangabes showed detectable iNKTs. So it's a challenging population to study. And then looking at 6p11, 6p11, we tended to see lower frequencies, but they were highly correlated. So in all future studies, we've just been using the unloaded and the PBS 57 loaded CD1D tetramer. We also went ahead and sorted these NKT cells in the initial studies and did some limiting dilution cloning, and then did the sequencing of some of these clones to really look at, again, the CDR3 alpha region, and we could show that this is, in fact, conserved and really like the pseudomangabe and rhesus macaque. It's just similar to humans and the mice, except for this lysine instead of arginine. So then I started looking at different species, because so far I'd looked in pseudomangabes, which is an African non-human primate species. So looking in Asian macaques, so the Indian rhesus macaque and then the Mauritian origin cynomolgus macaques. This was interesting that rhesus macaques, those were the hardest, that most of them had undetectable or below our limit, lower limit of detection. Whereas in Mauritian cynomolgus macaques, we invariably detected iNKTs that was circulating, and that ranged from 0.01 to about 1%. And this was very similar to what has been described in humans and in mice. So in the future studies that I'm showing you, the studies was restricted to the Mauritian origin cynomolgus macaques. So here, looking at these different species, it was interesting that their CD4, CD8 phenotype was very different. So the pseudomangabes, it's a natural host, and they didn't have any CD4 NKTs. So their NKTs were all either CD8 positive or double negative, whereas the Asian macaques had CD4 and CD8 iNKTs, as well as CD4, CD8 double positive iNKTs. So this was similar for the Asian macaques to humans, and in all these non-human primate species, it's different from mice in that mice don't have CD8 iNKTs. They have CD4 and double negative. So we think that in the natural host, this may be like an evolutionary way by which this iNKTs have evolved to sort of evade destruction by SIV infection. So we then looked at the function, and this function was done by one, like a non-specific function. If you just stimulate with mitogen, what cytokines are released, and then an NKT-specific function where we had this C1RD transfectants, C1R cells, stable transfectants of CD1D expressing C1R cells, and use that to load alpha-galactosylceramide and then stimulate the iNKTs. And basically showing that, there is some interspecies variability in that Sudi-Mangabe NKTs could produce IL-13, but not the Asian macaque species. And this was not because of any cross-reactivity, because with mitogen, it was all three species produced IL-13. So in the SIV negative setting, it seemed as if the Sudi-Mangabes had a different functionality. Sudi-Mangabe NKTs had a different functionality compared to the macaque NKT cells. And following SIV infection, this function was preserved, whereas SIV-infected rhesus macaque or cynomolgus macaques, they really lost all their, most of their interferon gamma function, and it was really predominantly IL-6. Also looked at the frequencies, what happens in Sudi-Mangabes, whether you look at SIV negative or SIV infected Sudi-Mangabes, the NKT frequencies and function are comparable. But in cynomolgus macaques, when you just look at the total circulating iNKTs, there's no difference. But when you look at CD4 NKTs or double positive NKTs, so basically CD4 expressing iNKTs are lost following SIV infection. So basically from these initial studies, we could see that there's preserved iNKT frequency and function in natural hosts of SIV, and there's a loss of CD4 iNKT and aberrant functionality of surviving iNKT in the non-natural host. And so, one of the questions is that, does NKT loss and hyperfunctionality contribute to aberrant immune activation and immunodeficiency in pathogenic SIV infection? So the next part of the studies is we briefly looked at NKT in tissues, because in SIV infection, in AIDS, we know that microbial dysbiosis is a huge driver of chronic immune activation. And then we also sought tools for in vivo NKT manipulation. So in mice, iNKTs are supposed to be really enriched in the liver, like they can account for 60 to 70% of that population. In humans, I think it's more in the gut, maybe some in the liver, and really nothing is known in the non-human primate species. So these are some unpublished studies where in monkeys, what we can do is we can biopsy. So we can get samples of the bronchoalveolar lavage, the bone marrow, rectal mucosa, subcutaneous fat, and lymph node. And so in a sort of sampling of, I think it's about 12 cynomolgus macaques, the frequencies in the blood and in the tissues really varied between animals. But on the whole, we didn't see any enrichment as such. Like the peripheral blood still seemed to be the higher. It was very comparable with bone marrow, but there was so much animal to animal variation. In one animal where we were able to get all the tissues at necropsy, really wanted to look at the liver. And really in the liver, you see NKTs, but it's not like what you see that they are enriched in mice. The surprising thing was in the fat, both omental fat and subcute fat, that they're really enriched. And there's a lot of literature out there where the NKTs are implicated in either driving obesity, I think there's all conflicting, or actually suppressing, preventing obesity and inflammation. We also looked at the gut because of my interest in AIDS, SIV infection, and the chemokine receptor profile of the iNKTs in the rectal mucosa is very different from that in the peripheral blood. So one is that over 80% of them are expressing more than at least more than one chemokine receptor co-expression, with a lot of them expressing CCR5 and CXCR3. And these are the ones which get lost. And so they could be contributing to iNKT dysfunction following depletion in HIV-SIV infection. Though in the setting of SIV infection, we don't have data to substantiate that. This is, again, in some animals, we were able to get enough rectal mucosal NKTs and PBMC NKTs to do some functional studies and basically show that with PMA stimulation there, not PMA, I think we did TCR stimulation with anti-CD3, that there is a different profile where the rectal mucosal NKTs are secreting more IL-2 and more IL-17. And even on CD1D alpha-gal stimulation on sorted iNKTs, we could see like a very different profile. So basically to say that, tissue iNKTs may be quite different from circulating NKTs. So for in vivo manipulation of iNKTs in murine studies and also in human studies, it's been the alpha-galactosyl ceramide, either soluble or pulsed on dendritic cells. And then from Stephen Porcelli's lab, like they're modifying these alpha-galactosyl ceramide analogs so they can be skewed to either Th1 or Th2. So at the time, one of my collaborators, Bob Shop, he was at NKT Therapeutics, and NKT Therapeutics was working on a humanized monoclonal antibody, which is really the 6B11 version, which was humanized and with high affinity. And the FC portion is an IgG1, so if it binds to iNKTs, it'll deplete them. But they were using this in phase 1 clinical trials, and so they didn't want to give that to me. But they had this NKTT320, which was an activating analog for which they didn't have any use at that time. So I was able to get an MTA for that. So NKTT320 and NKT120 are similar in terms of what they recognize. It's just that the FC portion is different. So the NKTT320 has an IgG4 FC. And so when you give this antibody, it'll bind to iNKTs, it'll activate them, but because this IgG4 FC will not bind to FC receptors, basically they don't get depleted. So, so this was then a study published in 2022, and the work done here was done by my postdoc at that time, Nell Bond. And basically what we did is we did both in vitro and in vivo studies in cynomolgus macaques and could show that in vivo NKTT320 really modulates the immune system and could have a potential as an immunotherapeutic tool. So some of these subsequent slides is just showing a few of the data from this paper that in vitro studies, when we stimulated with NKTT320, we could show that it's the iNKTs that get activated, but not the non-NKTs. And this is a short-term stimulation. And this was a dose-dependent response, where one of the things with iNKT stimulation is, especially at higher doses and for longer periods, is that the activated iNKTs, they start downregulating their T-cell receptor so that you almost start not detecting them. So this sort of effect could be two things. It could be that because the NKTT320 antibody is bound to the iNKTs, your tetramer is competing for the same site. So it's a masking effect, or it's a true downregulation. So that makes it sort of challenging to monitor iNKTs in vivo following NKTT320. So the way we got around by that is that baseline, in most of the animals that we looked at, most of the V-alpha-24 positive cells were iNKT. So we took the V-alpha-24 positives as a surrogate for iNKTs and the V-alpha-24 negative as for non-NKTs. And this is just to show that it's very specific for iNKTs, and the non-NKTs don't get activated within this a short period of stimulation. In vitro, because alpha-galactosylceramide is so much used, so we compared how these two activating reagents compare. And really, after 48 hours, they look very similar, but it's in the early that the NKTT320 activates, makes them polyfunctional much earlier than alpha-galactosylceramide. So in the in vivo studies, we were able to do PK studies with Bob Shaw because they did that for the NKTT120, and basically it's the same thing. And we tried 3 different doses. It was all IV, single IV doses. And basically, this shows like a saturation level of 6 microgram per mil, which was achieved with low, mid, or high dose, and for the most part sustained for about 7 days at least. And similar to our in vitro data, in vivo also we saw a similar thing, that it's a specific iNKT activation, and for CD4s more than CD8s. And the interesting thing was what happens later, that when we start looking at polyfunctionality, and this is all by mitogen stimulation, you see that initially it's only the iNKTs that become polyfunctional, but after about day 1 or day 3, you start seeing the other T cells also becoming more polyfunctional after NKTT320 administration. In the plasma, within 2 hours of the antibody being given, we saw this massive increase in different pro-inflammatory cytokines. And then we were able to do RNA-seq on bulk PBMC in 4 cynomolgus macaques, and really showed that within 30 minutes, like 104 genes get upregulated, and by 24 hours, and it's like a cascade. It's different genes getting upregulated. So what was interesting was there was a number of these genes which are markers for macrophage activation, T cell activation that got activated, and then saw these other genes also coming up which are implicated in resolution of inflammation. So schematically, what we were able to show here is that, it seems to NKTT320 has an effect on a number of different immune cells with both activation and genes implicated in activation as well as resolution of inflammation. So now I'm doing some funding for some pilot studies in 6 rhesus macaques that we are vaccinating with a DNA MVA AIDS vaccine with or without NKTT320 administration to see that could this be used as a vaccine adjuvant. Switching gears, and in the last maybe 10 minutes, I'll just touch on what is like the current active focus of research in my lab, and this is on rhesus CMV infection, on the placental transmission model of congenital CMV, and using this model to determine what determines whether a virus, whether a pathogen, in this case CMV, crosses the maternal-fetal interface to prevent, so that we can be informed on design of a maternal vaccine to prevent congenital CMV. So congenital diseases are really, like birth defects, are really common. 1 in 33 babies. This was from CDC. I think prior to January 20th, probably I haven't looked more recently. But for congenital CMV, 1 in 200 babies in the US are born with congenital CMV. Yet awareness of congenital CMV is really poor. So what this is showing is the percentage of women who have heard of the condition is this, whereas the actual problem is here. And this goes with what about congenital HIV/AIDS? People who have heard about HIV/AIDS is here, but the problem is actually minuscule. So this is really like a huge challenge in the field of congenital CMV, the unawareness about this problem. So it's the most common cause of congenital infection worldwide, and approximately 10% of those that are born with congenital CMV have severe sequelae at birth, And it's the leading infectious cause of childhood. So even if you're asymptomatic with congenital CMV, there is a chance of developing sensory neural hearing loss. And having a vaccine, a maternal vaccine to prevent congenital CMV has been like a tier 1 priority for a long time, but we don't have a vaccine as yet. And so what have been the challenges to develop a vaccine to prevent congenital CMV? One is just the biology of CMV infection, like all herpes virus infections, that natural immunity does not prevent reinfection. It can have a disease-sparing effect on outcome, but it doesn't prevent reinfection. So getting sterile protection, I don't think is realistic at all. So in the context of congenital CMV, what are the correlates of immune protection that would reduce risk of intrauterine or postnatal transmission and control CMV infection in infants? Is it antibodies? Is it T cells? Is it both? Probably both. What about the specificity, the quality, the location? I think location may be really important. And do we need something which is beyond natural immunity? Because natural immunity can be very robust. And so studies to investigate the determinants of CMV transmission and disease do require a relevant animal model of congenital CMV. So this is when I was at the New England Primate Center that I started collaborating with Sally Parmar, who's now at Weill Cornell. That time she was in Harvard. And to ask about, can we develop a rhesus macaque model for congenital human CMV infection? And so what we started is that at the New England Primate Research Center at that time, we had a cohort of CMV negative macaques, and which is very unusual. And so we were able to get pregnancies in this CMV negative, rhesus CMV negative dams. And we did these studies where we inoculated with 2 strains of rhesus CMV. And we did studies in immunocompetent dams as well as CD4 T cell depleted dams. Basically, in this study, we were able to show that rhesus CMV does cross the placenta, just like human CMV. And then in the context of CD4 depletion, you have 100% transmission and 80% fetal loss. But in an immunocompetent setting, you don't see that. So these studies were published in 2015, and we've gone on to show that if you pre-infuse hyperimmune globulin prior to inoculating with CMV, you can completely prevent congenital transmission even with the CD4 depletion setting. And then studies by Matilda have shown that natural immunity is partially protective, and also, maternal fetal immunity, which I'll just allude to in a few slides. So the model that we have is this, that we enroll rhesus CMV seronegative pregnant macaques. So at the primate center here, we have a colony that we call expanded SPF colony that is free of CMV, in addition to SIV, retroviruses, all the rest. And this is a really valuable resource so that natural pregnancies are enrolled into the study. At the end of the first trimester, we inoculate with CMV, and then we can do amniocentesis, ultrasound-guided amniocentesis weekly. We do an elective cesarean section near term gestation, so we can harvest the placenta as well as the fetal tissues, so we can really look at the maternal-fetal interface. So in this model, we can look at viral determinants, we can look at systemic maternal immunity, we can look at immunity at the maternal-fetal interface and see what determines whether the virus transmits from mother to child and what determines fetal infection. So this, I think she walked off, but this is, but this was a neat study from, during Matilda's graduate study period, where basically because worldwide congenital CMV is seen in the setting of CMV seropositive mothers, not CMV seronegative. And so this non-primary CMV infection the question is natural immunity even protective? And so she was able to do these studies in CD4 depletion rhesus macaques that was CMV seropositive, and could show that when CMV inoculation is done in the context of CMV inoculation and CD4 depletion is done in the context of primary infection, you see a viremia and 100% transmission. Whereas when you do the same thing in the context of a CMV seropositive macaque, you see like a blip of viremia, no transmission, 20% transmission instead of 100%, and no fetals. So that means the natural pre-existing humoral and CD8 T cell memory can protect even in the absence of CD4 T cells. This is also Matilda's work where we're looking at really immunity at the maternal fetal interface, because this is really important that, once we inoculate with rhesus CMV, whether or not it gets into the amniotic fluid, it could be determined at what is happening at the decidua, and then if it crosses the decidua in the placenta and into the fetus. And so in the maternal fetal interface, on the decidual side, we are looking at maternal tissue immunity. On the fetal side, we can look in fetal blood, in the placenta, cord blood, and fetal circulation. So one of the first things was to ask that, is monkey decidua representative of human decidua? And so this was really all Matilda's work, where heroically she collected all these decidua from uninfected, just normal pregnancies, and then from a collaborator with Zika infection, and basically developed a 28 color panel and showed for PBMC and decidua that these are very distinct populations in the PBMC and decidua, which reflect what is reported in humans. And then when you look across different trimesters, and we don't have that many animals across each trimester, but it does seem as if the first trimester decidua is quite different from third trimester decidua. And this is just like a fun schematic in one immunocompetent dam, where we are looking at the maternal PBMC, decidua, placental cells, cord blood, and fetal PBMC, and just showing with a panel focused on TB and NK cells what a different profile it is. What I found interesting is really looking at cord blood and fetal PBMC. The fetal blood we get from a cardiocentesis. So in humans, cord blood is taken as fetal blood. And for the most part, it's good to see that when we look at really fetal circulation cord blood, they look pretty similar. And this is with a DC mono NK panel showing that in the placenta, it's mainly all these tissue-resident macrophages. In the cord blood, it's mainly naive CD4 and CD8 T cells. So our ongoing studies are on looking at immune determinants of transmission, where currently we are doing a CD8 T cell depletion studies followed by primary infection to see what happens. And we have similar studies lined up for depleting NK cells, B cells. So that is one power of this model, that we can do these in vivo depletion studies and then really be able to look at in-depth in the tissues. The central question is there anything different in transmitters versus non-transmitters? And then defining viral and immune bottlenecks to transmission. So for these congenital CMV studies, we really have a very nice collaborative group with Oregon. All the virology is there with Sally and Weil at UC Davis with Peter Barry, and all the sequencing at UMass. Otherwise, in this, this is really a large collaborative work with folks in my lab, and really at the Primate Center, the veterinary medicine, pathology, and of course the Flow Core, which is led by Matilda. And this is, if you come to the Primate Center, we have in the center a memorial garden, and I really like the sculpture because it's dedicated in appreciation of non-human primate research, which significantly advanced medicine and the health. and welfare of human and animals. And this was set up by the previous director, who's no more, Dr. Andrew Lackner, who commissioned it from a sculpture friend of his in Arizona. Okay. That's it.
I think it's on.
Yeah.
Can you hear me?
Yes. I
Thank
can
you,
hear
Mindy.
Connie.
That was really very good. I had one question on your NKT cells in the gut, the functionality back on slide 26. Were those animal subjects SIV positive or SIV negative?
I'll have to go back and look, Connie, because these were just like few. I want to say that these were SIV negative, but I'm not 100% sure. We didn't have enough samples to look at SIV negative versus SIV positive. So in the same animal, we were just trying to, because when you get rectal mucosal pinch biopsies, most of the time, like when you isolate cells, you may just get 0.5 or 1 million cells, and that's not enough to do any NKT functional studies. So it would have been animals where we were able to get enough cells. And I'll have to go back to like the raw data to see which
I was
animals
just
these were.
curious.
Yeah.
Then if positive, what
Yeah.
the route of transmission was for infection.
Oh, so all our SIV infected monkeys are experimental infection. The pseudomangabes are all natural infection. And those are, so the Yerkes Primate Center has a colony of pseudomangabes. Most of those are naturally infected. There are very few SIV negative pseudomangabes. So early on, when I started my postdoc, we were able to experimentally infect them. So I was able to do some of the experimental infection studies. But then the USDA classified them as endangered, even though the pseudomangabes are not endangered. It's the red cap mangabey. But anyway, they were classified as endangered and we could no longer infect them. So after that, then my studies had to be confined to just the naturally infected pseudomangabes. For the macaques, they're all experimentally infected.
Okay,
Yeah.
it works. Very nice talk, Mindy. Thank you for that historical overview of everything. So regarding the NKT cells, there's been bodies of work to show that CD8 expressing cells, which would include NK cells as well as CD8 T cells, can have immunoregulatory functions, right, in suppressing inflammation during SIV infection. And there's some evidence to suggest that that is contact dependent in nature. Do you think that's what's happening with these iNKT cells as well? Or is it purely an endocrine effect from cytokine secretion?
So, that's the question as to thinking about iNKTs, how do they get activated, say, during SIV infection? So it's just models that people have said, because it can be either a TCR independent, which is cytokine driven. That would mean that something has to get activated, maybe dendritic cells, something which is producing IL-12, IL-15, which then activates. So that means iNKT activation is downstream of the initial DC or whatever activation. Or that it is like a danger signal, and so you have endogenous CD1D getting upregulated with self-lipids. And I don't know how to address that question. It would have to be some kind of, because we tried doing something like, how can we spatially look at iNKTs in vivo? So we tried by the tetramers, like if we take tissue, so we have to take fresh tissue and incubate it with CD1D tetramer, and which is conjugated to a fluorochrome, and then see by immunohistochemistry, just like in situ tetramer staining for CD8 T cells. But we were never successful. And I think because it's such a low frequency population, in mice, there have been some very elegant studies, that able to track iNKTs in vivo, showing spatially different things. But I've not been able to do anything